Yeah, dear Stefano, thank you very much for this very kind words of introduction. I probably would need some correction to what you were saying, but I don't do it. I'll leave it as it is. Thank you very much for the invitation to this wonderful place and this exciting meeting. I have to excuse my Italian is probably well enough to become fat during holidays in Italy, but it's not appropriate to give a scientific lecture. So I have to excuse that I'm talking to you in English now. My topic, or the topic I was given today, is DNA methylation. In the program, it's methylation, but I want to make clear we're talking DNA methylation, not on histone methylations or so. And I would give you a mixture between a bit of educational and a bit of data on what we are doing at the moment and what's the background of DNA methylation studies in malignant lymphomas. But before doing so, there are a couple of persons in the audience besides Stefano who spent some time in Kiel and know this man, Karl Lennart, who actually introduced the Kiel classification of lymphomas. And you know, all these kind of lectures nowadays are accompanied by quality control. So I leave you the question, what links the Kiel classification to DNA methylation, and to give you the task to think about this in the first part of the talk. That's a hand drawing of Karl Lennart, and that's a hand drawing in which he proposed his classification. I would say this is still essentially right. We have learned additional things, and I would like once in between come back to this and show you how we can integrate DNA methylation into this scheme too. But basically, I would like to talk with you on these following topics, some few basics and technologies, just to give you a background that we're all on the same page. Then I would like to introduce physiologic changes in DNA methylation, DNA methylation in lymphomas for determining cell of origin, DNA methylation in lymphoma pathogenesis, some models, DNA methylation and lymphoma prognosis, so can we use it, and a bit of visions with regard to etiology, liquid biopsies, targeted therapy, and classification. But let's start with the basics. So methylation of a DNA, modification of a DNA, usually takes place at CpG denucleotides. There is also methylation on other nucleotides, but that's the most frequent, and that's what we determine. You see the number of CG nucleotides in the genome is quite exhaustive, and around 70 or 80% are methylated. We can distinguish between methylation in or in front of genes and besides genes in intergenic regions. So the typical textbook view is that you have like promoter regions which can be methylated or unmethylated, some of them linked to CpG islands with a high density of CpGs. Then you have intronic sequences which can be methylated or not. And then there is a role of DNA methylation to silence repetitive sequencing that will not jump around into the genome. So that's a bit of textbook. Is this true? In part, it's true. So that's one of the studies we did already almost 10 years ago, in which we combined transcriptional profiling, so RNA expression by RNA sequencing, with DNA methylation. And this here, the y-axis shows you DNA methylation from 0 to 100%. And in red are the so-called absent transcripts, so genes not expressed. And in green are genes which are expressed, so it's a Burkitt cell line. And what you can see is that at the transcription start sites, those genes which are expressed have a low methylation, whereas those genes which are not expressed have a high methylation. So this textbook knowledge is right. High methylation leads to silencing, but only if it happens at the transcription start site. If you go across the gene further on, you see the situation changes. There is a higher methylation of a transcribed genes in the gene body. What's the function? It's not completely known. So one idea is that it's involved in splicing, for example. So if you talk on DNA methylation, you have to really talk on which part of the gene you are talking about. And if you really talk on silencing, you have to go to the transcription start site. Another site which is of interest are what I call the footprints of transcription factors. So the binding sites of transcription factors, that's the DNA methylation again from 0 to 100% across transcription factor binding sites across whole genomes. So behind this is like 20 genomes and all transcription binding sites of this given transcription factor. And what you can see, where the transcription factor binds, you have a much lower methylation. I always call this kind of footprints, but not footprints in the sand because they are not lost, but they are stabilized during further progression through the lineage. So you can keep this information as a kind of lineage determined for a certain cell. What are the techniques to detect DNA methylation? So we still do some restriction digest with DNA methylation dependent restriction enzymes. We do this in southern blots. I run a routine diagnostic lab. We do fragile X, and so you need to do this there. But it's rare. You can use antibodies to participate CpG-rich regions. That's a technique which has a pretty low resolution. I think it's not anymore that state-of-the-art nowadays. You can do bisulfite treatment and then therefore medical chemical modification. And nowadays you can also do direct sequencing using, for example, ONT technology. You need downstream analysis, which can be targeted sequencing, microarrays, or next-gen sequencing, depending on what you do here. So I would say our workhorses at the moment are more or less here in this region. And if you look into the development of technologies in the last years or decades, we started with single CpG loci. When I started DNA methylation, it was on imprinting disorders. It was back in the 1990s, middle of 1990s. I'm not so young anymore, Stefano, already some time ago. And then we got into this field of arrays of different companies, but at the end, predominantly Illumina arrays. At the moment, our real workhorse is the EPIC array, which works very good also on paraffin embedded tissues and so on. And you have the bisulfite pyrosequencing, which came and then was substituted more and more by bisulfite whole genome bisulfite sequencing or targeted bisulfite sequencing. And now the ONT is clearly coming because you can investigate both DNA modifications and sequence at the same time. So we have done full genomes here, first one, which look pretty good. The other advantage of some of these technologies is that at the same time you can interrogate genomic operations. For example, with these arrays, you can nicely produce a kind of array CGH profile and can look for operations. If you want to look for 11q lymphomas, that's mostly doable with a methylation in one step without an additional investigation. And also the bisulfite sequencing you can use, for example, here to show this homozygous deletion in the P16 CDKN2A locus. So you can combine genomic and DNA methylation analysis nicely in one step. DNA methylation is an active step, which is enzymized by DNMTs. Then you have 5-methylcytosine, but that's further modified in different directions. And we have to also make clear that many of the technologies we apply do not differentiate between classical 5-methylcytosine and, for example, hydroxymethylcytosine. That looks the same in the analysis. So if we talk on DNA methylation, we have to be clear that some of these probably rare other modifications we also detect and cannot distinguish clearly. We need special downstream analysis for this. And finally, pathologists around have to take care. You can also, on a global level, investigate methylation in a cell by antibody detection. And you see that there's a difference between 5-methylcytosine and hydroxymethylcytosine with regard to the expression, for example, in germinal centers and others. That's something probably underexplored yet, but it can be very nicely stained. That's something more for the pathologists in the room. Okay, that's a bit of basics. What about physiologic changes in DNA methylation? Please all remember that we all differentiate during our life. And you all are aware with B cell development, for example, from starting from a hematopoietic precursor, immature cells entering the germinal center, and you have plasma cells. So that's already a differentiation step which somehow needs to be controlled. And one controlling mechanism is DNA methylation. That's a study we did in the framework of the Blueprint Epigenome Consortium, a European consortium that was run by Iñaki Martin Zubero, my former PhD student and postdoc. And we investigated the DNA methylation across the undifferentiated cells into plasma cells. You see there's a change of overall DNA methylation. And if you do this kind of principal component analysis, you also see that the different populations clearly distinguish, so you can identify them. And if you do this in a more sophisticated point of view, then that's the differentiation from hematopoietic stem cells to bone marrow plasma cells. And these are so-called modules, and red is high methylation and blue is low methylation. And that what you can see is that the methylation changes, and that's a directed process. You never have such valleys, you lose methylation or gain methylation, but it's always the same direction. And by these modules, you can define the different steps of DNA methylation between the different differentiation stages. So it's directing the methylation in a direct order. It's this kind of footprints I've shown you before. And if you then associate this with function, then you see that those modules who lose DNA methylation are enriched in enhancer regions. So again, that's exactly the footprints I've shown you with valleys. So if there is an imprint set at this stage, it's maintained over the whole B cell lineage and so on. So it's really a directed, put in stone differentiation process. And you have some other genes which are increasingly methylated. And these are particularly genes which are polycomb targets in stem cells. So that's again differentiation genes which are anyhow switched off, most of them in a normal cell, and which are silenced completely. So there's a directed order of DNA methylation, and you can use these modules to define a kind of cell of origin here already, and can say, okay, that's most likely looking like a precursor B cell or a germinal center B cell or bone marrow plasma cell. So if you put this together, you have demethylation at enhancers, which are genes in the immune system, B cell transcription factors, and you have demethylation of heterochromatin, and you have methylation of polycomb targets. So it's a kind of really structured process in the normal B cell development. And particularly this here you can use for further analysis. So what is this telling us about Carl Lennart? You have thought about this? Okay, it's pretty easy. You have here a differentiation step, which we can now really demonstrate in demethylation. And also Carl Lennart differentiated between genes which are off, like CD5, and on. And this is also imprinted in the DNA. We can look into the DNA. And there's one additional thing which we haven't talked about yet, but which I think is personally is most fascinating. He also drew this kind of morphology of a chromatin. And I've shown, you see here, these are the nucleoli in centroblasts, which are outside, and the immunoblasts, which looks like a mirror egg, we would say in German. And this kind is what you pathologists look on it. It's the morphology. And this guy, also working in Kiel, Walter Fleming, he coined the expression, it's the chromatin, the stainable part of a nucleus. And that's nothing else than epigenome. It's nothing else than DNA methylation-driven epigenome. And it's chromosomal location. And this guy also coined the name of germinal centers. So I think he has to be really put aside here with Lennart. But if you now look into a microscope, think about you're looking at the epigenome there. Okay, so what I've shown you, we can use diagnostically, we can determine cell of origin. And this is a kind of pedigree, yeah? It's a kind of phylogenetic tree from a stem cell to a differentiated tree on the end. The same you can do with DNA methylation. Here are the precursors. Here are the mature B cells, germinal center cells, plasma cells. So you have a differentiation from here to here. And we, for example, looked where are acute lymphoblastic leukemias are located. They are here. So they are immature. Where are Burkitt lymphomas located? They are here. And what about lymphomas with an immature phenotype and a Burkitt translocation? They are here. they are in the ALLs, at least a subset of those which are really ALLs. So you can use this to really pin down the cell of origin. I show you another example which is not yet published. It's the study of T prolymphocytic leukemia. So you know in the peripheral blood, the cells look like mature T cells, but the translocation takes, or the inversion 13 driving the TCL1 gene takes place in a thymocyte. That's something you can show by DNA analysis. And again, here are the precursors, here are the mature T cell populations. And what you can really see is that the TPLLs also originate from a step in between. They are definitely not mature T cells. They are more immature cells, though they appear in the peripheral blood with a few stains you do probably 4 or 5 markers as mature cells. So that's something we can learn about by DNA methylation. What about DNA methylation in lymphoma pathogenesis? I could tell you hundreds of examples of given genes which are switched on and off in TPLL and so on. I would like to go to my favorite, which is Burkitt. So we did a study in which we investigated the whole genome bisulfite sequencing of Burkitt lymphomas compared with to follicular lymphomas. You can see that a correlation with gene expression you find only in a very subset of cases of CPGs. And we defined so-called differentially methylated regions which correlate with the transcription between follicular and Burkitt's. Don't think about the numbers, it's a huge number. And we were particularly interested in the function of TCF3. That's a transcription factor which is activated in Burkitt lymphomas, either directly through mutations or by mutation of ID3, which is much more common, which inhibits TCF3. So we see in like 60 to 70% of the Burkitt lymphomas mutations in this gene. Another way of deregulation is differential splicing. So we wondered, has this something to do with DNA methylation? The answer is yes. So if you have a high TCF3 expression, then the target genes of TCF3 are demethylated. That means that the TCF3 footprints across the genome are switched on. It's one of the highly significant genes, but even more, TCF3 regulates itself through differentially methylated regions within the gene. So it's a kind of auto loop of DNA regulation via DNA methylation footprints. And it also regulates other key players here like SMARCA4. And if you put all this complicated experimental data together, you come up with a very easy model. In normal B cells, TCF3 is inhibited by ID3, and TCF3 induces ID3, so that's a regulatory loop which leads to a hemostasis. You have a differentially methylated region here. The target genes of TCF3 are unmethylated, and these again, among them is SMARCA4, and that leads to the expression of target genes. What happens in case of translocations, or in case of activation of TCF3, sorry, not translocations? You have either ID3 mutations or TCF3 mutations. You have a strong demethylation of this differentially methylated region. So that's part of, it's like a green traffic light. It allows you to give transcription now, a lot of transcription. So this traffic light, which is here probably yellow, is here green. So a lot of transcription going on. And it also switches on all the target gene traffic lights to yellow, to green. So it's all going on. One of them being SMARCA4, but SMARCA4's target genes are bound but not switched on. That's the pathogenesis of Burkitt lymphoma. I think that's a very core, one of the very core mechanisms there. And we can do we can do a huge number of these kind of stories. We have just as we're talking, we have submitted a paper on expression in Hodgkin's lymphoma, in ALK positive lymphomas, where we show how this methylation is involved in regulation of certain genes. So you can associate it with lost B cell identity in Hodgkin's lymphoma with DNA methylation. You can identify the transcription factor driving this, and so forth and so forth. So there are numerous studies coming out from this. I would like to stay with Burkitt once briefly, Because if you look into the literature, you will see that ID3 mutations are common in sporadic Burkitt lymphomas, EBV negative, that's what we showed. But they seem to be much rarer in endemic Burkitt lymphomas. And another list of genes are, I would say typical Burkitt genes are not mutated so frequently in EBV positive Burkitt lymphomas. And that's a study we did with several, like Professor Leoncini in the room, but also others, where we looked for DNA methylation of these genes. It's now being written up, the paper, and it shows clearly that these genes have a hypermethylation. So we think that in sporadic Burkitt lymphomas, these genes are mutated, and in endemic Burkitt lymphomas, they are hypermethylated and thereby silenced. It's the same effect, essentially. And why is this the case? Because EBV positive Burkitt lymphomas has a strong hypermethylation phenotype. The same holds true for gastric cancer, EBV associated. So you see here the overall DNA methylation across all CPGs. And what you can see really is that endemic Burkitt is really outstanding here. It's extremely high and showing this hypermethylation. So the same holds true in other virus-driven diseases. In EBV positive gastric cancer, it's a bit different signature, but that's probably one of the things viruses are doing. And you have to consider that DNA methylation is also involved in detection of foreign DNA in cells. So that's probably one of the mechanisms behind this. DNA methylation and lymphoma prognosis. I would like to show you here one example. I have to say that's a critical topic. If you look back in the literature like 10, 15 years ago, you see quite a number of papers investigating MGMT methylation, P16 methylation, P53 methylation, and correlating with this prognosis. All these papers do not take into account the physiological DNA methylation, and they do not correlate with clinical data and with treatment and with multivariate analysis. We did, a PhD student of mine did a lot of work to trying to reproduce this, and it was all not reproducible. So it's unfortunately never yet published, but it's hundreds of cases, clinical trials, we couldn't verify many of these data in diffuse large B-cell lymphoma. Where it really holds true is in CLL, where you can identify different groups based on DNA methylation. That's the N-CLLs and the M-CLLs and the intermediate group. I don't want to go into the biology behind this. I want to show you that's important from the diagnostic point of view, that you can reproduce this array data. Though I have to say the array is meanwhile routine in our laboratory. We run this like once a week, at least once a fortnight. So the array is cheap, fast, solidly working. But if you want to break it down to a more common technology, you can do pyrosequencing, and you can break it down to bisulfite pyrosequencing. And we did a study between Barcelona and Kiel at that time, comparing the results. And that was a very, I would say, a very sophisticated design, which was essentially had the idea to also do technology validation and see how comparable the results are. And you see there's a Cohen's kappa of 0.9, which is exceptionally good for a kappa for bisulfite analysis in both laboratories. So that's a quite valid technology. The correlation is well between the both laboratories, and you can clearly identify clinically relevant subgroups here, which obviously have some correlation to IG mutation status and others. And this also refers to treatment. And you can validate this in independent series. That's a very robust thing. That's CLL, that's probably easy. But as said, in other lymphomas, it's definitely more difficult to do. So at the end, let me give you some visions about where I do see DNA methylation in the future, in addition to what I've shown you. And I've taken a few samples. But let's first think about this. DNA methylation is not like the genome. You have one genome and probably a tumor genome. DNA methylation changes according to sex. So you all know already that females have two X chromosomes. One is inactivated by DNA methylation. That's easy. Ethnicity, we can clearly identify ethnicity based on DNA methylation. Age, sorry to say so, by aging you change your DNA methylome. Cell types, I've shown you with disease states and so on. So there's a lot of moral heterogeneity going on. And what you can investigate is also environmental factors addressing this. So you don't need to ask who is smoking from you. If you give us a blood sample, I will tell you. We are able to do this. So what we have done is we have looked into all our lymphomas we had, whether there's a smoking signature and whether smoking could affect lymphoma pathogenesis. I have to say that doesn't work. It probably works better in myeloid diseases because the signature is more myeloid signature. But you can, in principle, look into this etiology. You can. What we also looked is into the age of the biological age of a patient and the calculated age of the tumors. Things like this. That's really very fancy analysis. Gives you a very nice impression on what's going on. Another example I've shown you is EBV signatures, so viral infection as a driver that we can nicely identify. Selina Glaser in my lab has identified predictor, and what we can simply do if we run a methylation analysis, we do not know only the methylation, the classification, the chromosomal imbalances, because we're calculating, we can also tell you whether this is EBV positive or negative. So that's all in one shot. You can also investigate the EBV, for example, itself, whether it's methylated or not, because there are tumors which methylate EBV itself or not. This is a tumor cell line which has a high methylation of EBV virus DNA itself. Okay, if you can do this all on a tumor, can you do it on liquid biopsies? Yes, you can. You can run DNA methylation analysis on liquid biopsies. Actually, it's a test which myself as a clinical geneticist use like daily because it's in Germany run for the detection of trisomy 21 in maternal blood. But it's doable also on liquid biopsies. You can at the same time distinguish genetic aberrations and DNA methylation alterations. You can also say what's the amount of DNA from your tumor as compared to other organs, because if you have a patient with an organ failure, you can identify which is the origin of the organ. And Ola Amapol in our institute, Professor of epigenetics, we are working on lung cancer, can nicely predict in the liquid biopsies what kind of lung cancer you have in front of you without any biopsy and any genomic sequencing there. So that's all doable on liquid biopsies too. And then there's the question, what about treatment? Obviously, you can change epigenetic marks. There is a widely used treatment in myeloid disorders, azacitidine. You might question whether azacitidine is really working about epigenetics. It does work about epigenetics, but it's toxic in addition. So there might be other ways of doing this. But that's a kind of general theme. I don't know whether I would be keen on having my whole genome demethylated. So I would probably have more targeted therapy. And there are options. You can, by CRISPR technologies or TALENs, you can either switch on methylation and by this silence a gene, or you can, by TET, for example, you can remove methylation and by this activate a gene. And actually, Susanne Benz in my group, together with Albert Jelsch, they are going one step further. They address mutated alleles and try to deposit methylation in mutated allele. Think about a RAS mutation activating and you can silence it by methylation. That's probably, and only the mutated allele, not anything else. So that's something we work on. That's kind of allele-specific or mutation-specific DNA methylation. And finally, what about classification? So this is from brain tumors. And this is from sarcomas. And my lab is also running a diagnostic lab for trials in pediatric tumors, pediatric brain tumors, particularly atypical tertiary rhabdoid tumors, where we run with bisulfite, with arrays and classify the patients accordingly. In the new blue book of the WHO, this is already part of a classification in brain tumors. The sarcomas have been published recently. It works nicely. You can simply run an array, put it in their database, you get out the correct diagnosis, and if not, you ask your pathologist to look at it, and then you can discuss which better. Will lead to some discussions. We have them already. But this is a kind of classification which is really working robustly. And the advantage is it's not so sensitive like RNA expression because the DNA is more stable. You can do it in all samples. It works, DNA methylation. It's very robust to detect. I have to say, I doubt it will come up in lymphomas pretty soon because we probably have the advantage that we are far more advanced. We have already like 50 to 60 lymphoma entities, and they started with one. So what we can see differences between CLL and TPLL and so on, but it's much more difficult to diagnose differences between follicular lymphoma and diffuse large B-cell lymphoma entities. I'm not sure we will go into this direction, but we anyhow have discussions on the WHO. So we will see whether this classification is earlier than the next classification of the WHO. That's what I wanted to share with you. I think there's an enormous potential of DNA methylation. It's not a new thing. We think about DNA methylation since 20 or 30 years, but the technologies are there now to measure this. It's a very robust technology. I haven't shown ONT data, but the ONT data is really exciting because at one read, you can identify all the different methylation changes. So that might in addition help, but for ONT technology, you need brilliant DNA you never have. So that's only exceptionally possible. But for people who did all the work, I have to particularly name Ole Ammerpohl, who's the leader of the epigenetics group in our institute, Selina Glaser, Laura Wiele, our bioinformatician, Selene Kretzmer, the group in Heidelberg, and Iñaki Martin Zubero and Henk Stannenberg in the Blueprint Consortium. And the good thing on epigenetics and DNA methylation is the following. If you don't know an answer to a question, you can say it's all due to epigenetics. Thank you for your attention.
Reiner, thank you very much for this comprehensive really lecture on methylation. We have some minutes. This is an invited lecture, so usually there is no discussion. But if there are questions, I think that Reiner will be happy to answer.
Thank you very much. Wonderful. I'm really convinced that you have reason because recently we revised a small series about 50 patients with the LBCL that we previously characterized by nanostring for cell of origin. And we measured the polycomb gene expression. And in particular, we saw that the expression of BMI1 that previously we checked in myeloid diseases was prognostic. So what do you think? I saw that you mentioned the polycomb genes in your presentation. What do you think about the role of BM1 as a H2 or other polycomb genes in follicular lymphoma or the LBCL? Thank you.
I think there's a clear role. So another example is EZH2, which we know it's mutated or activated. And there is a clear role for polycomb complex in the germinal center reaction. And let's say we see different distributions of mutations, now coming to the genomic side, in histone modifiers between different subtypes of lymphomas. The germinal centers have much more mutations of several histone modifiers than others do. I have to say we are not at the end of this. So while we're talking, we have submitted a manuscript on KMT4C mutations in inactivating mutations in different diseases, including Hodgkin's lymphoma. So I'm absolutely sure that there is a role on this epigenetic level, but it's much more hard to measure because it's much more difficult to do histone methylation changes in paraffin embedded tissue. On the other end, you can see the downstream effects of this on DNA methylation. So I'm convinced it will have a more predominant role in the routine praxis than histone modifications. But I fully agree, there is a role of histones in also prognostication.
Okay, Reiner, congratulations, of course, for this great lecture. I have a curiosity. You mentioned during your talk about the methylation-induced viral presence. So I was curious to know whether you're aware of the same phenomenon with bacterial infection.
With bacteria?
Yes.
Oh, that's a good topic. No, we haven't done this systematically. So what we have done is we have explored our genome data, our whole genome data, for bacterial DNA in the samples we have sequenced. But there was nothing conclusive. We know that, for example, Helicobacter can induce DNA methylation changes. There is data on this. But let's say take the gastrointestinal microbiome, for example, how this alters the DNA methylome is not really explored. Actually, my most quoted immunology paper is a mouse model showing that T cells change the DNA methylation upon microbial exposure in mouse models. So that's clearly, there is an effect, but it's not that well measured yet.
I would have a couple of questions, but I will discuss with you this topic later on personal grounds, since we should be strict to the times. So